we're looking at cell division. Decided to bring this after genetics because there's something I really want you guys to pay attention to. I'm sure cell division is not new, right? There are two methods of cell division. The first one is known as mitosis, and the second one is known as meiosis. Okay, so for today, I will talk about mitosis. Then we can talk about meiosis later. But I can also say some few things about it. Okay, let's move on. What is mitosis? Mitosis is an additional division. What do I mean by that? When a cell divides by mitosis, it produces two of its own. Mitosis can be likened to cloning. What do I mean by cloning? You know, when you make a clone, we're making an exact copy of an organism. Like you're producing itself, another one. So you're making double of the organism. So if a cell replicate or divide via mitosis or mitotically, it's going to produce itself. Everything exactly the same thing. Okay, let's move on. Cell cycle. Most cells in an organism go through a cycle of growth, development, and division called the cell cycle. Okay, now, interface. Please stay with me. Before a cell can start dividing via mitosis, it has to prepare itself to go into that process. And that preparation now, the stage is what we know as interface. Please stay with me. Interface. So what happens at interface? Okay, this is the period during which the cell cycle of a cell is the period during the cell cycle of a cell's growth and development. A cell spends most of its life in interface. Please stay with me. In case you see this in a JAMB question, if they ask you that, in which stage now do you think a cell spends most of its life? It is interface. I'll tell you why, because there are three different stages of interface. Now, okay, let's move on. The first stage is called the G1. G1 means the first gap phase, or the gap 1 phase. The first gap phase. It is the period of growth. Remember, if an organism is going to divide mitotically, right, the organism would have a nucleus normally. So the first thing that would happen is that the nucleus must replicate. This nucleus will now become 2. Are you guys my point now? Okay, now let's look at it. This stage lasts longer than other stages. A cell goes and carry out its normal functions. It goes and it carries out its normal functions. Then the next stage of interface is called the S phase. S phase means the synthetic phase. It is the stage at which some vital organs are being synthesized. Now, a cell continues to grow and copies its DNA. Please look at this. It copies its DNA. It becomes 2. Nucleus. Are you there, please? It copies its DNA. Now, there are 2 identical strands of DNA. All of the 2 of them will now look so much alike. Then, sister chromatid, that is 2 identical chromatids, centromere, what hold that sister chromatids together? What are we talking about? When you talk about centromere, like something that is holding, when you have a chromosome like this and it's splitting, like it's split, there's something that is attaching to it, that attaches to it to pull them aside. Are you getting my point now? Okay, all of that happen at the synthetic phase. Okay, then the next phase is the gap 2 or the second gap stage. The cell is ready for mitosis. The cell stores energy that will be used during the mitotic phase. Okay, please stay with me. There are 4 stages of mitosis. There are 4 stages of mitosis, and I want you to pay attention. Please, I am sure you must have done this, but let this remind you of what you have done. Okay, the first stage is known as the prophase. Remember, I did not start with interface because interface is not one of the stages of mitosis. Interface is just the preparatory stage. It's just the stage that prepares the cell to go into what? Into reproduction. Okay, we have the prophase, we have the metaphase, then we have the anaphase, and the telophase. So you may as well try to remember PMAT. Exactly now. Okay, so we try to look at what happened at each of them. Okay, mitosis. The nucleus and its content divide. Cells divide to increase their number. Remember I told you that in the case of mitosis, the cell will split to become what? Two. The cell divide to produce two haploid daughter cells. What do I mean by haploid? That means the cell will now produce what? Two haploid daughter cells. Okay. Now, let's move on. Sorry. Now, then the phases of mitosis. Prophase. Copied DNA condenses into chromosome. The nucleolus disappears and the nuclear membrane breaks down. Spindle fibers begin to form. Now, everything we're talking about here will happen at this stage. You can still see the chromosomes here appearing distinctly, right? But they will soon start condensing, where it will now look like a thread, so that it will be able to move into the process proper. Now the next stage is metaphase. I put this M here, M for middle. I'm going to explain the meaning of this. Chromosome line up in a single file at the center. That center is the middle. So if they are giving you, what JAMB will give you is to give you diagrams of all of this. And I think this metaphase is one of the most important stages. One of the most important stages that they use. Now, you see the chromosomes lined up at the middle. Once you see the chromosomes at the middle like this, know that it is what? Metaphase. M for middle, M for metaphase. Okay. Now, the next one, anaphase. At the previous stage, all the chromosomes were here, right? Remember that spindle fiber attached to each one. It attached to each one. They were like this before. So what the spindle fiber will do is to help you pull these guys to this side, pull this one to this side. So look at it here. They are already splitting. Are you getting my point? So the stage at which this happens is anaphase. Now let's look at what is there. Sister chromatids separate. The spindle fibers begin to shorten. Why? It begins to move to become smaller. Why? Because it would now be pulling the sister chromatids apart. Okay, now let's move on. Last stage is telophase, right? A nuclear membrane forms around the chromatin. Now, chromosomes begin to unwind. Spindle fibers break down, two identical cells form. Let me help you. Let me give you what I use to explain this. All these four stages, I can break them into two. I break them into what we call karyokinesis and cytokinesis. But of course, I always explain this after I must have explained the first one. Okay, karyo means nucleus. Cyto means cytoplasm. Kinesis means division. So when you hear karyokinesis, remember that before that cell, before it started dividing, what happened here? The nucleus doubled. Right? The nucleus doubled. Where's the S phase? Yes, I said where is it? I said copies with what? DNA. Can you see it now? The nucleus now became two. Right? Okay, so let's go back. Okay. Karyokinesis, which is the nucleus. So the nucleus will have to become two. After that has happened, then cyto is cytoplasm. This is the cell here. The nucleus has now become two. What you have here is the cytoplasm. What else do you need? The cytoplasm must split. It will split. It will split. It will split. It will split. Before you know it now, it split into two. So you have two new cells. Can you see that? Hello guys, is it clear? Okay, now let's move on. Result of cell division. It results in two cells. Daughter cells are genetically identical. They are the same. That was why I said it's like you making clones. You're making clones. But this kind of cloning now is natural. Yes, it's natural. Although we can make artificial clones, we won't go there today. Okay. Now, daughter cells are genetically identical. They are the same thing, which is a direct opposite of what we have in meiosis. In the case of meiosis, the daughter cells do not identical. They are not identical. These guys, they are identical. here in meiosis, they are not identical. Please, I hope you guys are still following me. Now, mitosis will produce 2 daughter cells. Meiosis will produce 4. Are you there? We produce 4 daughter cells. The daughter cells in mitosis are diploid in nature. Diploid. The daughter cells in meiosis are haploid in nature. Are you getting my point now? This stage is longer than this stage. This one is not, it's just 1 step. This guy is not, it's like 2. Okay, so let's look at what we have here again. Allow multicellular organism to go and develop. Of course, this mitosis occurs in the somatic cells. It is the process by which our blood cells increase in number. The nerve cells increase in number. Every other cell increase in number. But in the case of meiosis, it has to do with the sex cells. That's the reason why it must be haploid. So that if the sex cell coming from one is coming to meet with another one, it becomes diploid. Okay, what is again? In the case of mitosis, mitosis does not in any way support genetic variation. Are you getting my point now? But this guy is a tool for genetic variation. It's a tool for genetic variation. Why? Because it is not completely the same thing. At the end of meiosis, the daughter cells are not, they do not resemble each other. And they don't even look like their parents. Okay. Now, this mitosis helps us to repair and replace our worn out or damaged cells. Any cell that is died, that is spoiled in the body, they are being fixed via mitosis. Now, it is also critical for repairing damages. Example, bone breaks. Cell division will not produce new bone that patches the broken piece. Okay. Now let's look at questions here. DNA in eukaryotic cells is contained. Please quote me. They said eukaryotic, not prokaryotic. That was where we started from. DNA in eukaryotic cells is contained in we have central vacuole, we have nucleus, we have lysosome and Golgi body. It is contained in the nucleus. Although prokaryotic too would have their own genetic material, but it is swimming freely in cytoplasm. It doesn't have an enclosure. Okay. Now, number 12. A man who is heterozygous for the disease hemophilia. Let's go there. A man who is heterozygous for the disease hemophilia, stay with me. You might decide to use any other alphabet that you like, but let me use, let's say, I'm using H now for hemophilia. If you know the meaning of hemophilia, see, this is disease of the what? Of the blood. So anybody who's hemophilia now would have it. It will be so difficult for them to produce blood clots. So it's a little wound like this, they just keep bleeding. Okay. Then they said the man is heterozygous. So this man is like this. Right? The man is like this. Then they said marries a woman who is double recessive for hemophilia. Double recessive. So this is the man, this is the woman. I hope you're there. So the meaning is that the small h is the hemophilia. The hemophilia code. Right? Okay. Now the point is this. They say what percentage of their offspring will have the disease? I want you to pay attention to me. If you cannot interpret genetic questions, you can't get the questions right. Interpretation is very, very key. You should be able to interpret it. Now we have been able to do that. The man is heterozygous, that is why it is here. The woman is what? Double recessive. This is to scatter your head a bit, to confuse you. Double recessive, which means she is homozygous recessive. And that is what you have there. What percentage of their offspring would have the disease? Obviously, this woman, since she's double recessive, then the small h is the hemophilia trait. Okay, so let's do it. So this guy and this guy, right? We have this. Do you agree with me? Then this guy and this guy too. We have this. Then this guy and this guy, obviously hemophilia. Then this one too, right? So let's say these two will be what? Hemophilia, right? So that will now be, since it is 2/4, it will be 50%. Let's check. What's the answer? 50%. You're correct. This is JAMB question 2019. So let's take a look at this question. It says a man who is heterozygous for the disease hemophilia marries a woman who is double recessive for the hemophilia. Okay, now let's try to interpret this. The man is heterozygous, so heterozygous, their names are different, right? He now married a woman who is double recessive. Double. So the woman is homozygous recessive. Are you there? Okay. Now the question is, what percentage of their offspring would have the disease? Okay. So let's look at it. Let's do this cross now. Obviously, since the woman is double recessive for the hemophilia, this is hemophilia. This guy. Right. Okay. So let's solve this. We have this. Right? We have this. We have this. We have this. So, if you are to interpret their genotypes, these guys are the same thing, these guys are the same thing. So, we have 2 ratio 2. These guys are hemophilia. Okay, so that will give us what? 2/4, which is what? 50%. I don't know where this one came from. The offspring are 4. Okay, so our answer is 50%. So I hope that is clear. Please, you can't why ask question like this. Okay, please go back to when I was talking about mitosis and meiosis. I used the word cytokinesis. I said it's a division of what? Of the cytoplasm. Okay, now this question says that cytokinesis of mitosis is a process that ensures that daughter cells get the necessary organelles. B, there is distribution of a complete set of genes into each daughter cell. Then daughter cell inherits new genetic combinations. Then worn out organelles are excluded from daughter cells. Now let's look at them one by one. Daughter cells get the necessary organelles. There is distribution of a complete set of genes into each daughter cell. During cytokinesis, the cytoplasm is being divided, right? Now, organelles are complete. Like what is here is also what is here. Everything must be complete. Are you there now? So the right answer for that question is B. There is distribution of a complete set of genes into each daughter cell. Okay. Number 14. A group of closely related organisms capable of interbreeding to produce the fertile offspring are known as member of kingdom, class, family, and species. The answer is species. I'm going to explain something to you. Now, we have, when you talk about classification of organisms now, I'm sure you must have been taught classification in SS1. Okay. Now, what are the hierarchies for classification? Let me just chip in this. You might need this. Now, we have what we call King Philip came over for good spaghetti. Okay, and let's look at it. The king means kingdom. The Philip means phylum. Came is class. Over is order. For family. The good is genus. The spaghetti is species. Now, this is the lowest cadre of classification. It's the lowest cadre of classification, and for organism to be able to produce fertile offspring, please quote me, they must belong to the same species. Please stay with me. We have cousins everywhere, like I used to tell my students. We have some of them that are in the jungles, the lions, the elephants, the monkeys, all of them are cousins, right? Why? Because they belong to the same kingdom with us. They belong to the same kingdom, Animalia with all of us. But they do not belong to our species, so we cannot reproduce together. Now, you must have heard of liger, right? Where some scientists came together to breed lion and tiger together. Yes, they have an offspring, but liger cannot meet a liger to produce another liger. It doesn't work. So their offspring is not fertile. Are you getting me now? It is not fertile. A dog can just do like mate with something else. We've heard of similar cases here and there now, right? But the offspring of that union can never be fertile, which means that it cannot reproduce. Why? Remember I talked about chromosomes. All of them have different number of chromosomes. They have chromosomal number that differs. Somebody who is having 40 and someone who is having 30. If you split to produce gamete, this will be 20, this will be 15. Are they not supposed to meet? They are supposed to pair. If they pair now, there will be excess 5. That will cause problem. Let's keep that. Okay, next question. Number 15. The cytoplasm of a cell is considered a very important component because option A, it regulates the amount of energy in the cell. It suspends all cell organelles. It is the outermost part of the cell and it is solely responsible for cell division. Let's look at it again. It regulates the amount of energy. Told you cytoplasm has nothing to do with energy. If you talk about energy, then mitochondria comes first to your mind. Then it suspends all cell. When they say suspend, it means that it is in this cytoplasm that all the other organelles are suspended. Are you getting my point now? All the other organelles are suspended in cytoplasm. So the cytoplasm is like a fluid. The other organelles are there. They are there. It's just like an open, like in a room now, like on your table, where you have your books, you have your bag, you have your writing materials. So the surface of the table is the cytoplasm. All those other stuff that you keep on it are like the organelles. So the answer is C. It suspends all cell organelles. The mitochondria is found in it. Golgi body, lysosome, vacuole, all of them are there. Okay, now look at this. The overall reaction in glycolysis can be summarized as, now let's talk a little here about glycolysis. Okay, almost. Okay, now when you talk about respiration, what comes to your mind is eating of food, breathing in of oxygen and turning out carbon dioxide, but it goes beyond that. Respiration is the breaking down of food that you have eaten, of nutrients that you have in you to produce energy. And the process by which that is done, the first process is called glycolysis. And let's look at it. We have glycolysis. Can we split it? We have lysis and glyco. Are you getting me now? This glyco is from the word glucose, right? It's from the word glucose. This lysis is from the word lyse. When you lyse something, it's like you spoil the nature of that thing. Let me give you an instance. If you put sugar on hot pan, what happened to the sugar? It becomes watery. That is what happened to that glucose in glycolysis. So that it's going to expose the content of the glucose, then you'll be able to absorb everything you need from it. Okay, so this glycolysis now, when it happens, the glucose will continue to be keeps being degraded rather until it gets to what we call pyruvate. Pyruvate, which is a 3-carbon compound. But of course, we're going to have 2 of it. 3 carbon, 2 of it. Okay, let's look at the option. Option A, we have pyruvate. 1 pyruvate in option A. No, it's not 1 pyruvate. Why do we have 3 carbon here? Please stay with me. The glucose you are starting with has 6 carbon. So how are you going to end with 3 carbon? What happened to the remaining 3? So it's going to become 2. So these 2 now, these 3 carbon in 2 places, making how many carbon? 6 carbon. So that explains it. So that is why it can never be A. A is wrong. Now let's look at option B. We have 2 pyruvate plus 4 hydrogen plus 2 ATP. Now let me tell you one thing. These 2, option C and D, are wrong because of ADP. You cannot have ADP, you can only have ATP. ADP means adenosine diphosphate. But what the ultimate aim of this glycolysis is to produce energy. What is energy? It is in form of ATP. So it can only be this or this. But we have been able to fault option A. The only thing left is option B. It can't be this ADP. Why? Let me tell you one thing. When you talk about ATP now, it is when you have phosphate plus phosphate plus phosphate. Then those three plus what we call adenosine. That's the reason why the full meaning of ATP is adenosine triphosphate, 3-phosphate. Am I with you, please? So this energy that is here is stored in between the phosphate. So when you now use an ATP, which of course is energy, when it is used up, something will be released. That stuff that is released is called ADP. ADP is energy what? Empty. There's no energy inside it. So it can't be this. So the answer is B. Is that taken now? Please, I want to advise you that you can go over this again and again and again and again. I've chosen not to talk about some topics that you should be able to go through. You should be able to go through yourself. But these are some of the topics that I know that students run away from. They run away from genetics, the hybrid, monohybrid, we've talked about that. We talked about mitosis, right? And I also explained meiosis to you. We talked about cell division, right? We might also, maybe later, talk about transport system, where we'll be able to look at blood circulation from systemic circulation to pulmonary circulation and the likes. So I wish you guys the very best in your exam.